Okay, welcome to lecture 5. Um, in today's lecture, we'll be discussing something called Bayes' rule, and we'll also be discussing independent assortment. So when we talked about conditional probability, I'll remind you that sometimes you want to calculate an outcome, okay, and the outcome depends on a condition. In this particular case, you took P's that have this particular genotype, you cross them with this genotype, you have an F1, and then you select some round P's, and you don't know if they're big R, or big R, little r, so we put an R dash. And then we want to say if you just pick one of those at random, and then you self it, what is the probability that the progeny of that selfing are round, right? And the way you would figure that out, you might want to remind yourself, is this going to be figured out with A, B, C, or D, right? Which one of those represents the law of total probability? Okay, well, the correct answer is D, because the way that you would do that calculation, you would say, what would be the probability of obtaining a big R dash outcome given that you picked this particular parent, right, times the probability that you in fact picked that particular parent, and you would add that to the probability of having a round outcome having picked this parent, so the heterozygote genotype, times the probability that you picked the heterozygote genotype. And then we'll just fill these numbers in here. So this will be probability of having picked the big R individual is 1/3. The probability of having picked the big R little r individual is 2/3. And then if you did pick the homozygote, then this would be guaranteed to be round, so that would be 1. And then if you did pick the heterozygote, then the probability of obtaining a round would be 3/4. So the whole thing would be 1 times 1/3 plus 3/4 times 2/3. Okay, so that was how we did conditional probability. But you can imagine you might find yourself in this type of situation, which would be a little bit inverted or a little bit different. All right. Now, suppose you actually do pick this particular genotype, the phenotype. You pick a round pea plant, and then you self it. And we're not going to ask what the probability of a particular outcome is. We're going to say you observed that outcome. All right. So you self that plant here, whose genotype you don't know, but you know their phenotype. Right. And when you self that, you collect 4 plants, and 4 of those plants, and they all produce round peas. Okay. So then you might be able to ask a slightly inverted question, which is, what is the probability that the plant you picked was big R, little r? Now you might say to yourself, well, maybe it's a little less likely than 2/3. And the reason you might say it's a little less likely than 2/3 is you might say, well, if I did in fact pick the heterozygote, which had a 2/3 chance, maybe I would have expected to at least get 1 wrinkled pea plant from that outcome, but instead I actually got 4 rounds. Now, that's still fairly likely having you picked a heterozygote, but you might say to yourself, well, maybe it's not much less likely that I picked a heterozygote, but maybe it's a little less likely. And then you could ask even further, what if I, in fact, instead of selfing and yielding 4 round pea plants, what if I actually obtained, say, 1,000 pea plants from that, and every single one of those was round, right? Then you might say to yourself, well, maybe the chance that I actually did pick the heterozygote is fairly low now, because that outcome would be very unlikely. And so to actually put a number to this, and by putting a number to this, you either did or didn't pick a genotype. So it's a little bit weird to say what's the probability that you picked a particular genotype. That's a little strange. The way you would think about this is you would imagine if you were to bet, okay, how would you calculate the probabilities for a strategy of betting? All right. You don't know for sure which one it is, but you can estimate with some likelihood the maximum probability. All right, and so we're going to actually talk a little bit more about this type of inverted type of conditional probability. Before, okay, we talked about the probability of an outcome, right? So you have some outcome that you're trying to figure out what the probability is, given that something happened. And so the way we would say this is would say, what's the probability of having picked 4 round pea plants, given that a big R, little r individual selfed? All right, that was the way we've been doing conditional probability. But this type of inverted probability is a little bit different, because what you're actually saying here now is, what's the probability of something actually having happened? Right? What's the probability of the condition now given the outcome? So in this particular case, instead of saying what's the probability of getting 4 round pea plants given that this particular genotype selfed, it's actually saying what's the probability that it was that particular genotype that selfed given that there are 4 round pea plants. And this gets to something called Bayes' rule or Bayes' theorem. And I'm actually going to do some stuff on the board to walk through how we would think about this. All right. And so I'm going to actually switch over now to Zoom. Whoops. And we're going to do this on the board now. Now, what I want to show you to actually get you to think about how this works and how you understand Bayes' rule is I actually have 2 buckets here. I have a red bucket and I have a green bucket. All right. Now, in the red bucket, I have a bunch of vials that have been labeled red. This may not come up really well on the screen, but there's 9 vials that are labeled red here, or pinkish, and then there's 1 vial that's labeled green. Okay, so if you look closely in there, there's 9 vials that are labeled reddish, and then there's 1 that's labeled green. Okay. Now, in the green bucket, I have the other way around. Right? In the green bucket, I have 9 vials that are green and 1 vial that is sort of a red-pink color. All right. Now I have these 2 buckets, right? And so what I can do is I can, go over here and I can draw one from a bucket. All right. So I'm going to draw one, and you didn't see which one I drew from, okay? And what I picked was this one here. All right. So this is a reddish pinkish color. So which bucket do you think did I pick it from? Okay. Well, probably most people would say that I picked it from the red bucket. Now, that's possible. Actually, in this particular case, that's the one I did pick it from. I did pick it from the red bucket. Now, I could have picked it from the other one, but that chance would be pretty low, right? So here's the green one. There's a small chance I could have, in fact, picked the red one from the green bucket, but it's a smaller chance of having picked there. So Bayes' rule gets at this type of thing. Okay. Now, what we want to be able to do is you say, okay, before there was maybe a 50-50 chance of having picked one of those, right? And what we call that probability without any information is a 50-50 chance. In this particular case, we say there's a 50-50 chance. That's the prior probability, all right? So prior to me telling you any information about it, there's a 50-50 chance. That's the prior probability. But then once I pull something out, then I have a new probability of having picked, in fact, the red bucket. It's no longer a 50-50 chance anymore, because I think, well, maybe it's more likely than 50-50 that I, in fact, picked from the red bucket. Okay, so remember, we talked about probability, conditional probability, and we'd say that the probability that A and the probability of B, the probability that both happen, all right, that A and B both happen, can be written in 2 different ways, okay? It can be written as the probability of A and B both happening. It can be written as the probability of A, right, given B happened, times the probability that B in fact happened. So basically you have this sort of first thing happened, B happens first, and then after B happens, then A happens, given this condition B. So that would be equivalent to saying the probability of A and B. All right? Now, you could also make that equivalent to another way of thinking about it, where you say, okay, well, instead of B happening, well, let's say A, in fact, happened first, all right? So the probability that A happened, all right, which is independent of B, all A would be given that way. These two things are equal, all right? The probability of A given B times the probability of B is equal to the probability of B given A times the probability of A. All right, well, that's actually kind of interesting, right? Let's actually set them up equal to each other on one row, because we can do some interesting manipulation. All right, so I'm going to say the probability of, I'm actually going to use a different marker. I'm going to use black. I'm going to say the probability of A given B times the probability of B is equal to the probability of B given A times the probability of A. Is that how I want to do that? Yes, that's how I want to do that. Okay. Now, we can do a little bit of algebra. We can pull this down, right? We can pull this down over here, and we can rewrite that as the probability of A given B is equal to the probability of B given A times the probability of A, right? All over the probability of B. Now, I'm going to erase what's on top because I'm actually going to get into a little bit of what's underneath here. So this part underneath is kind of interesting here, right? This is the probability of just B happening, and it has nothing to do with whether A happened or not. Now, we can decompose the probability of B using the law of total probability, all right? We can say, if we want to break it down into either A happening or not, using the law of total probability, law of total probability, we can say that the probability of B is equal to the probability of B given A having happened times the probability of A having happened plus the probability of B having happened given A not happened. All right, so we can actually say not A times the probability of not A. Sometimes I put a minus there, right? So you could just put a minus there if you want. So that's the law of, that's going back. The probability of getting cancer, lung cancer, is the probability that you get lung cancer given that you're a smoker times the probability that you're a smoker, plus the probability that you get lung cancer given that you're not a smoker times the probability that you're not a smoker. One thing you can actually break this down, because A and not, remember, A and not A, right? Those two things are mutually exclusive. And they add to 1. The probability of A plus the probability of not A is equal to 1. All right? And that's just a reminder of, like, what you can put here. You could decompose this into it further, right? You could say, for example, that the probability of A plus the probability of, let's call it C, plus the probability of D. We could say that that's equal to 1, and they're all mutually exclusive. Let's suppose they're all mutually exclusive. You can break down the law of total probability further. We've done things like this already. You could say, for example, that the probability of B would be equal to the probability of B given A times the probability of A plus the probability of B given, let's say C now, times the probability of C plus the probability of B given D times the probability of D. All right, we said that A, C, and D are mutually exclusive. All right, so, and then they add to 1. All right, so you can break that out however you want. Keep this in mind because you'll kind of want to think about this under some circumstances, how you can decompose B into, multiple sums when there are multiple mutually exclusive outcomes that you're mutually exclusive events. But we're gonna keep it simple for now, and we're just gonna focus on this framework, which is what we've been mostly using for class. All right, now that's what this here is, right? Now that's interesting because what that means is that this and this, or the form that I just showed you with, A and C and D or any other form, right, can be plugged into the bottom of this, all right? So let's think about what that does. So let's erase probability of B here, and we're going to replace that with this, all right, which is the probability of B given A times the probability that A happened plus the probability of B given, I'll use the minus sign for not A, times the probability of not A. Okay, now that looks like a bit of a bear, all right? But what I'm going to show you in a moment is that this is going to be very helpful because sometimes this is hard to know, but we can easily figure out all these other ones. All right, so sometimes you don't know what the probability of A given B is, but you can figure out the probability of B given A. I'm going to cut to the chase for a moment, but then I'm going to kind of backtrack a little bit. Sometimes we know the probability of an outcome given a condition, but we don't know the probability of a condition given an outcome. All right? So it's a little strange, and I'm going to walk you through going back using the buckets now as an example. All right. So that's Bayes' theorem right here. Okay? This is Bayes' theorem. Sometimes I'll call it Bayes' rule, but it's the same. All right? Right down here. Okay? And just to be clear, this is one form of it in which I've broken up, A and not A, right? I've broken the probability B into those separate mutually exclusive things, but it could be broken up in any way. It could be, A and C and D with the sum of all of those conditional probabilities. So it could be done that way. So what the heck is all this about? How is this useful? Okay, well, let's go back to the buckets. All right, so I'm going to keep that down at the bottom there. Right, and I'm going to actually transpose this. And what we're going to ask is, what's the probability that I picked? Let me figure out which one I have been doing. The probability that I picked the red bucket, all right, given I have a red one, all right? Okay, so the way we're going to figure this out is we're going to say the probability, all right, that I picked red given, all right, I should say red bucket, right? The probability that I picked from the red bucket. All right, let's be clear about that. All right. Given I picked a red vial, so I'm just going to call that red vial. Okay. So this is going to be basically A here, and this is going to be B. Right? The probability that I picked from the red bucket, given that I picked a red vial, all right, is going to be equal to, all right. So now A and B are switched up here, and that's kind of interesting because we'll be able to use that because this is going to be an easier thing to calculate. So the thing that here is now it's going to be the probability that I picked a red vial. I'm going to write picked red vial, just to be as clear as possible. All right, probability that I picked the red vial given that I, picked the red bucket. All right, well, that seems easy, right? So if I did, in fact, pick the red bucket, what's the probability that I picked a red vial? Well, it's 9 out of 10, right? Because there's 10 in there, and 9 of them are the reddish pinkish, right? So just a reminder, so we're kind of doing this funny switcheroo here. So this is B, right? And this is A, and so we want the probability of A. So the probability that I picked the red bucket, right? Picked red bucket. Now, This is the probability that I, in fact, picked the red bucket without any information about what vial I obtained. What did we call that before? Well, we said this is the prior probability, and that's going to be 50-50, right? It's 50% chance that I picked this one, 50% chance for that one, right? This here is called the prior. All right, prior means before. Before I did anything, I have these probabilities. Then I have an outcome, and my probabilities are going to be updated. The updated probability will actually be called the posterior. All right, but this is going to be A here, right? The probability that I picked the red bucket. Okay, now that is going to be all over. Okay, that's going to be now here. Now a thing that's useful to remember here is oftentimes you'll see a scenario where, what's here is the same as what's here, right? So probability of B given A times the probability of A. That's the same as up here. Probability of B given A times the probability of A. So that's the same, right? This part here on top is the same as that part. All right, so this is going to be the probability, again, this part that I picked the red vial times, so that's B, right? Given the probability that I picked the red bucket, right, times the probability that I picked the red bucket. Picked red bucket. All right. So, okay, it'll be okay. I can fit the rest of this on the board. Okay, so, and that's going to actually be added to, now below is just the probability of B, right? But I'm now under these two conditions. The probability that I picked the red vial, okay? The probability that I picked the red vial under the condition of picking the red bucket, and it's going to be added to the probability, here's B there, the probability that I picked the red vial, given, now, not that I picked the red bucket, this is not A now. It's in fact that I picked the green bucket, all right? So the probability of actually that outcome given the alternative condition. The alternative condition, which is mutually exclusive, because I either pick this one or this one. So it's going to be the probability that I picked the red vial, given that I picked the green bucket now. All right, and that's going to be multiplied by, right, the probability that in fact, I should say that that's not A right there. That's a typo. All right, so this is given this condition of not A. Probability of B given not A times the probability of not A. Probability of picking the red vial given picking the green bucket times the probability that I picked the green bucket. Picked green bucket. All right, and since I left some gaps here, all right, this is B and this is A, right? Picking the red bucket, and then this is A here, right? This pick red bucket, and then this is B, but now it's not A, right there, and then this is not A. And that's the format right here. Okay, now the reason we go through all of this is it's hard to know, what this probability is. I picked a red vial, okay, and you think I picked the red bucket, but, maybe I picked this one, right? But there's a 50-50 chance starting out, but now what's the probability? And when I say what's the probability, it's like, what's your betting strategy? If someone doesn't know how to calculate this, if you know how to calculate this, you will win in a game of gambling with them. All right, so let's plug these numbers in. All right, so I'm going to erase this because it's getting a little bit messy here. Okay, the probability that I picked the red vial, given that I picked from the red bucket, all right? Well, that's this one here, and it's 9 out of 10, right? 9 out of 10 for right there. All right? And then, that's given I picked. And okay, what's the probability I picked the red bucket? That's the prior, right? So that's 1/2. Right? That's right there. Right, those are multiplied by each other on top. And then down here, it's the same thing, right? It's 9/10 probability that I picked the red vial, given that I picked the red bucket. And then this right here is a half. Okay. Okay, well, what about this? Okay, so now we have the probability of having picked the red vial, given that I picked from the green bucket. So what's that? Well, I picked from this green bucket, and there's only 1 out of 10, so that's 1/10, right? Excuse me, 1/10, not 1/9. All right, now what's the probability that I picked the green bucket? Well, it's 50-50 for these, so it's not always going to be that way, but in this particular case, it's 50-50. So this is 1/2. Okay, let's plug these numbers in a little bit more neatly. This whole thing is going to be 9/10 times 1/2 all over, again, it's the same thing, 9/10 in this case times 1/2 added to 1/10 times 1/2. All right, and if you reduce that out, that whole thing, actually the halves reduce, all the tens reduce out, and then the whole thing becomes 9/10. That's actually kind of interesting. So wait, what did we calculate? Always ask yourself that question. Well, now that's the probability that I picked from the red bucket. Now you might be saying to yourself, well, it's either, you either did or didn't. So how's weird to say probability? But that's how it works. And then again, that's why I keep referring to, like, if you were to bet based on those probabilities, you would win a game if someone wasn't betting using that framework. All right, 9/10. And kind of, in this particular case, it kind of makes some intuitive sense. Okay, so now we're going to actually walk through this as an example with some P's. All right, so I'm going to go back to the PowerPoint here, and then we'll walk through and actually do some genetics. Okay, so in this particular question here, we asked before, okay, you picked one of those round plants and you yielded 4 pea plants. What is the probability that you in fact picked the big R little r genotype? Okay, so you want to go to use Bayes' rule for that, all right? And we'll just, and I'll, walk through this one. It's a little weird. I mean, I'll admit Bayes' rule is kind of strange. One thing I do want to say is Bayes' rule is used all the time, right? So spam filters, for example, that for your email, they just sort of say like, okay, what's the probability that this is spam? And it's like, oh, there's like 5 sketchy terms, and so it evaluates all the spam that's being put out there, and it says, oh, this is spam, and this is not spam, and it calculates, oh, what's the probability that this is spam given this like list of sketchy terms, and if it's above some threshold, it's like, oh, it's spam. Artificial intelligence uses Bayes' rule all the time. Okay, so we're going to go to this, and we say I'm going to write this on the board before I get off. So we put this one and big R dash, all right, and we get 4 round. And what's the probability that we picked the big R little r genotype? I wanted to write this on the board while you're not looking so that when I get rid of this slide, we have it with me still. All right, so voila, there we are. All right, we selected this one. We observed 4 round P's, and what's the probability that we in fact picked big R little r? Okay, so just a reminder, Bayes' rule, okay, is the probability of A given B is equal to the probability of B given A times the probability of A, right, all over the probability of B given A times the probability of A plus the probability of B given not A times the probability of not A. Okay, so how do we put this into the framework? So that's Bayes' theorem right there. I just want to keep a record over here so I don't mess this up. So these are just some notes for myself. just so I remember like what the exact framework is over this. Is notes for me. Okay, so so what is A? All right, so in this particular case, what we're going to do is we're going to put this in the framework. This is going to be basically we're calculating what's the probability, and that's the question here that we picked big R, little r. All right, what's the probability that we picked the big R, little r genotype? All right, so this is going to be A. Right, given this outcome. Right. So what's the probability that some condition has happened given an outcome, and the outcome was that we got 4 round. All right, that's what we're interested in. But it's hard to calculate. But we can say the other way around. We can say, what's a probability that I obtain 4 round given the big R, little r? Right, we can already calculate that. Right, so we can put that up here. So this is going to be B, right, and then this will become, right, the probability that I obtained 4 round, right, that's B, that's that outcome there, right, given this condition that I picked the big R, little r, times the probability that I in fact picked the big R, little r. Okay? And that's going to be all over, and then it's going to be the same. The probability that I obtained 4 round, given that I picked the big R, little r, times the probability that I in fact picked the big R, little r. And that's going to be added to, right, the probability that I got this outcome that we observed 4 round. But now, it's big A, sorry, this is B, this is A, and this is A here. Picked big R, little r. This is B, right? This is A, this is A, right? So the probability of B, which is 4 round, now this is not A, right? So having picked not big R, little r, right? But big R, right? Times the probability that I in fact picked that one, right? Because I remember I picked the round one, and I picked round. I didn't know which genotype it was. It was big R, little r. So there's, 2 ways. So now the probability that I picked big R, right? And so this is B, and this is not A, and this is not A. All right. So I'm going to erase Bayes' rule here, right? And I'm going to actually start adding some numbers into this. Okay. So we're going to kind of start here, and we're going to work forth. So the probability that I picked the big R, little r genotype, given that I got 4 round as an outcome, is going to be equal to, and I'm going to start plugging the numbers in here. Okay. If we picked the big R, little r genotype, what was the probability that I would obtain 4 round? Okay. Now remember, if big R, little r selfs, right? This is going back to the Punnett square. The chance that I get a round is 3/4. But I'm asking about 4 rounds. All right. So then that is 3/4 times 3/4, right? The first one, the second one, the third one, the fourth one. So it would be 3/4 to the 4th power. I just want to check my notes here and make sure that I'm working okay. Okay, 3/4 to the 4th power. All right. And then what's the probability I picked the big R, little r genotype? Well, we said that was 2/3. Right. That's always 2/3 when we've been talking about that one a lot. All right. Now that's going to be all over. All right. Now the probability that I picked 4 round or obtained 4 round, given that I picked, this is the same as before, so same as above, 3/4 to the 4th times 2/3 plus, okay, and it's going to be, okay, so what's this now? Okay, so here, this is the alternate outcome. This is A, I picked big R, little r. Now, not A is I picked the homozygote, right? So if I pick the homozygote, what's the probability that I obtain 4 round pea-bearing plants? Well, it's guaranteed, right? Because there's no way to get any wrinkled. So it's 1 times 1, it's just 1, right? You could say 1 to the 4th if you want, just to kind of keep it consistent with before. And what's in fact the probability that I in fact pick the homozygote is 1/3. Okay? And actually, I don't have that calculation in front of me right now. But it'll be worth calculating it out. So I'm actually going to do that right now. Give me a second. I'm going to do that on the board here with the calculator because I don't want to go and try to reduce that live. So 3/4 to the 4th. So 0.75 to the 4th is equal to, now you could leave it reduced if you want, but in this particular case I want to actually calculate this out. So this is 0.316 times 2/3, so I'll just do times 0.66666666, alright, which is equal to 0.211 up on top, and then over here, so that's 0.211 again, right? And then this is just a third, so plus 0.33333, right? So I'm going to calculate that, plus 0.3333333, okay, and then I'm going to do make sure I'm doing something right here. I feel like I made a mistake. Okay, that makes sense. And then this here is 0.211. Okay, sorry. And then this here is a third, so 0.333. Okay, so yeah, I made a mistake on the thing. So it's going to be 0.211 plus 0.33333, which is about 0.54. Okay, and then so I think I can do a 1 over. Okay, 1 over times 0.211 about. Okay, so that's equal to 0.387. All right. So that's kind of interesting, right? So with the probability that I in fact picked the big R, little r, before I told you anything, it was 2/3, right? But now it's actually gone down a fair bit, right? It's gone down to 0.387. So what happened is that based on this outcome, my probability for that has changed, right? And so if I got more and more and more rounds, right? If I got like, I picked, a round P and then I selfed it and I got like 100 rounds, right? Then it's going to approach very low a very low number that I picked a heterozygote. Okay, so that is Bayes' rule, and we'll work through some more problems with that. Okay, so the next little bit is we're returning to Mendel now, and we're going to actually talk not about one single gene, but we're going to talk about 2 different genes, and in this particular case, they're going to be on separate chromosomes. And the reason and the significance of them being on separate chromosomes is that the segregation of one set of alleles from a given gene is going to be independent from the segregation of the alleles of the other at the other locus, right? So you can take 2 P's. This particular kind is round and green. Okay, so as we've been knowing, as we've been talking about, wrinkled is the round is dominant. Okay, so we have a round P, and it's green, and we haven't talked about green versus yellow much yet. But in this particular case, green is recessive and yellow is dominant. So round is dominant, and wrinkled is recessive, and yellow is dominant, and green is recessive. Okay, so this gives us We have a pea plant that's big R, which makes it round, and little y, which makes it green, right? And we cross it with a pea plant that is yellow and wrinkled. It's wrinkled because it's little r, and it's yellow because it's big Y. And we cross those together, right? And because big R is dominant, we end up with a round pea bearing plant, and it's also yellow, all right? And so, but we also, both the round locus and the yellow locus are heterozygous. So what happens in the next generation? So the important point here is that the segregation of big R with little r is not going to influence the segregation of big Y with little y. Now the importance of that, like that they're independent, remember that if we're trying to calculate, the probability of one outcome and another outcome, like the probability that the first child is a boy and the second child is a boy, if they're independent, you just multiply those probabilities. So if there's a 50-50 chance for big R, right, and there's a 50-50 chance for big Y, then any particular gamete that's produced by this individual in a selfing can be calculated using that principle of independent probability. So you could say, okay, what's the probability of obtaining a big R, big Y gamete from that individual? Well, there's a 50% chance for that, big R. There's a 50% chance for that, big Y. So 1/2 times 1/2, a quarter. So there's a 1/4 chance that you obtain a big R, big Y gamete. Likewise, there's a 1/4 chance that you obtain a little r, little y gamete. Likewise, there's a 1/4 chance of a big R, little y gamete, and a 1/4 chance of a little r, big Y gamete. Now, that's because the segregation at this locus doesn't influence the segregation of that locus. So, when we're talking about how we predict phenotypes in the next generation, remember, the Punnett square is a representation of fertilization probabilities. So you have on the top row the probabilities of one set of gamete types, and on the left column, on the leftmost side, you have the probabilities of the gametes coming from the other sex. And I've been in this class doing male pollen on the top row and then the ovules on the left. So we're going to actually do that probability of these fertilization events, and we're going to walk through the Punnett square. But now there are 1, 2, 3, 4 possible gamete genotypes instead of 2 possible gamete genotypes because of the fact that there are 2 genes. Okay, so now in the next slide, I'm going to show you what that Punnett square looks like. Oh, sorry. I'm going to back up. Before I get to what that Punnett square looks like, I'm going to tell you right now that the ratio that one observes is a famous ratio of 9:3 to 3:1 of these different genotypes. All right, the most common phenotype, phenotype. The most common phenotype is going to be the round yellow, and that's because of dominance, because the round allele and the yellow allele are dominant. All right, the least common phenotype is going to be the phenotype associated with homozygosity for the 2 pairs of recessive alleles. All right. So wrinkled in green, and that is at frequency 1 relative to 9. And then the alternating one locus is recessive phenotype, and then the other locus is the dominant phenotype. And then the inverse of that, where it's the alternating locus, so is going to be 3 and 3. So we have round versus green, right? Remember, green is recessive and round is dominant. And then we have wrinkled, which is recessive, and yellow, which is dominant. Okay, so there's this famous ratio, the 9:3 to 3:1 ratio, and where that comes from is what I was expecting on this next slide, but is coming from the Punnett square with 4 alleles in this situation. Okay, so here again we have these individuals selfing, and this is what I was trying to bring us to. On the top row, okay, we have the frequencies of the male gametes, right? And so the segregation pattern of R doesn't influence or associate with the segregation pattern of Y. So what you end up with is a 1/4 chance of big R big Y, 1/2 times 1/2, a 1/4 chance of big R little y, right? So on and so forth. So 1/4, 1/4, 1/4, 1/4. Those are the probabilities of the four gamete types. And then we get the same probabilities right here. Now, what you can notice here is in this column here, right? And in this column here, this row here, is that we have all of the round yellows, right? And that's because when you inherit this gamete, no matter what gamete you inherit through the male lineage, when the female gamete is this genotype, the female gamete has the two dominant alleles. And so whatever is inherited through the pollen is not relevant, no matter what, because the female gamete has the two dominant alleles, going to be round yellow, round yellow, round yellow, round yellow. And the same logic applies here. Round yellow, round yellow, round yellow, round yellow. So no matter what this gamete genotype is, this gamete genotype from the male is determining it. So you end up with 1, 2, 3, 4, 5, 6, 7. All right. Now you can see that there are 16 possible combinations. Now the genotypes will of course collapse. When because you'll get genotypes that are equivalent depending on what the alleles are coming from, either the female or the male, and so you get 7 in that way, all right. But then in this particular case, you have big R and little y coming from dad, and then you have the inverse, little r and big Y coming from the female gamete. Now you inherit a big R allele and a big Y allele, and you're a double heterozygote. So that again produces the round yellow. Same logic applies over here. Big R, little r, big Y. Again, you are a double heterozygote, round and yellow. So that's the 7, 8, and 9 ways in which you can have a round yellow phenotype. All right. Now notice, obviously the genotypes are all going to be different, but as long as you're heterozygous or homozygous for the wild type allele at both different loci, you will be round and yellow. Okay, let's look at this kind of awkward situation here in which little r, little y, and little r, little y, I guess it wouldn't be considered awkward, but it would be considered rare. This is the very rare circumstance where the recessive allele is inherited for both genes through both gametes. All right, so here is the 1/16th chance that you end up being wrinkled. Now, looking at these ones, the 1, 2, 3, the round yellow, in this particular case, this is a scenario in which you're big R, or big R, little r, right? So that means that you're round no matter what, but the little y allele is coming from dad, little y here and little y here, right? And the little y allele is also coming from mom, right? So that produces the green phenotypes. Those are the 3, right? So there's 1, 2, 3 ways, and that's the same way that the wrinkled yellow ones are produced. There's 3 ways for that to occur, in which the little r allele is inherited from both the male side and the female side. Little r, little r, little r, little r, little r, little r. But there's a among that group, right? There's a 3/4 chance. Actually, if you look at this here, this is almost like a mini Punnett square in which everybody is little r, and there's a 3/4 chance among that subset that you're either big Y, little y, right? These two here, or big Y, right? So inside here actually is kind of like a mini Punnett square, all right, where everybody has inherited. Everybody has inherited the little r, the 2 little r alleles that are recessive, making them wrinkled. Okay, so it's worth thinking about this particular cross here. This is a cross not with selfing, but is a different particular cross. This is in which a heterozygote, which might have a 1/16th chance in this particular configuration, is crossed with a homozygote. This is like a test cross, right? Because this individual here, you don't necessarily know the genotype, but here we're going to say it's this particular genotype, it's a double heterozygote, is crossed with this recessive, so wrinkled. So let's take a moment to think about what the ratio of the progeny would be in that particular case. How would you do that? I'm going to give you a second to think about it, and then I'm going to walk through it. Okay, now the way I would do that on the board is I would actually do that sort of thing that we call the Punnett rectangle, all right? In which, because this genotype, little r, little y, all right, is being crossed with this genotype, all right, this individual is only going to produce gametes of one type, all right? And so you can do that, what I would call the Punnett rectangle here. All right, in which all of the gametes, there's a probability of one. The probability of one up here. All right, all of the gametes are going to be little r, little y. All right, so then you have all of the four possible combinations, each with a 1/4 chance. So you can be big R, big Y. You can be little r, big Y. You can be big R, little y. And then you can have the little r, little y. All right. So let's just walk through the genotypes that would end up here. This genotype is going to be 1/4 of the time, 1/4 of the time for that one, 1/4 of the time for that one, 1/4 of the time for that one. You can already see that it's probably not going to be a 9-3 to 3-1. It looks maybe like it might be a 1-1 to 1-1. Okay, so what are these ones? Okay, so big R, little r, colon, all right, and then big Y, little y. All right, so let's talk about the phenotype here, all right? This allele produces the round, and this allele produces yellow, so these are going to be round and yellow. All right, so this is little r and little r, all right, and then big Y and then little y. So this is going to be wrinkled and yellow again. All right, so now we inherit the big R, so big R, little y now, right? So this is going to be round and green, and then this one's going to be little r, little y, and so this one will be wrinkled and green. All right, so those are the phenotypes that we would observe, and they're going to be in a 1-to-1-to-1-to-1 ratio because each of these has a 1/4 chance. Now, it's worth thinking about what happens in meiosis in this particular case. So just to walk through how that works, when you have an individual that is heterozygous for both, right, we're going to show here, and we're going to use a little bit of color. We're going to use, let's see if we can get some holiday colors going here. Okay, good. All right. So with respect to green, I don't have yellow, but with respect to green, so you're going to start off, and this is going to be for DNA replication, right? And so we're going to have, this will be the green versus yellow, right? Okay, so this is before, I'm going to draw, let me get back to the zoom here, all right? So that's fine, all right? So what I have up here, okay, is this is before DNA replication, and I have the chromosomes, and this is going to be with respect to round and wrinkled. right? The alleles there, and then this is going to be with respect to yellow versus green. Okay, so I'm heterozygous, right? And haven't done DNA replication yet. But what I will now do is mark the genotypes. And so because I'm heterozygous here, I'm big R here and little r right there, right? And then with respect to yellow and green, I am big Y right here and then little y right here, right? So this is before DNA replication. So DNA replication, now the sister chromatids will become genetically identical to one another, right? So then this is going to be also big R, and then this one here is going to also be little r. And then over here it's going to be, so I'm going to draw it a little differently here. There we go. Right, big Y over here, get a little bigger, right? And then over here, it's going to be a little y. I'm going to erase this. Okay. Now so this is after DNA replication, and now the chromosomes are going to split apart from each other. Now remember, in the first division of meiosis, the chromosomes go part ways from each other, but the chromatids don't split. All right. So in the first division in M1, what's going to happen is, this pair of chromosomes will go this way or that way, right? And then, one configuration would be to go this way and this way, right? So when that happens, then what you'll end up with is sort of a preliminary, you'll end up with this configure over here, right? And then the genotypes, the way I have it here is big R, and then big Y, and big Y, right? So that means that over on the other side, I get the opposite of that. I'm going to have little r, and then I'm going to have little y, right? Now, you have to make sure you be able to remember that that's how the segregation patterns are going to happen. But we'll be 50% of the time with this genotypic configuration. I'll get to that in a second. But it's important that you don't show the sister chromatids coming apart this way. This is called the divisional segregation, where the alleles segregate away from each other, but the sister chromatids do not. Now, what happens here now in the second round of meiosis? Well, that's the sort of like the mitotic division, and that's when the sister chromatids split apart. So this will split, this will split, this will split, this will split, and what you'll end up with is gametes that are going to be, in this particular case, big R, and big Y, and big Y. Okay? So, these are genetically identical gametes the way that this configuration has occurred. There isn't any crossing over. We'll get to that later. Now, half the gametes will have this configuration, how this occurs, and then it will be the other way around here. So then it will be, and I'm just going to draw, sort of little squiggles. Okay, and now this is little r, right? Because there's only the little r, right? And then there's little y and little y. All right. Now you might be saying like, okay, but how do you get the other? This is like 4 meiotic products. Big R, big Y, little r, little y. How do you get the other ones? How do you get big R with little y, right? Or the other way around. And the point here, and this is this point about independent segregation, is this is one way that they lined up. But there's a 50% chance that they line up the other way, and that this goes this way, and this chromosome goes that way. I mean, you could just switch the way they line up. Now, that configuration now, big R is now segregating with little y in the first round of meiosis. So then we would have big R, little y, big R, little y. And then over here, it would be also switched around, in which big Y is segregating with little r. So this would be big Y, this would be big Y, and then so we'd have little r with the big Y and little r with the big Y. And the point here is that, how this segregation pattern happens is independent of this segregation pattern. And so it's important to think about how this is working, these probabilities, in the context of meiosis. All right? But just to summarize, what you end up with, because of that independent pattern of segregation, is in that scenario where you're heterozygous, you end up with, these big R, big Y, big R, little y, little r, big Y, and then little r, little y. Each of those is going to be 1/4 of the time. And the reason they're 1/4 of the time is half the meioses will have one pair of configurations, and the other half of the meioses will have the other pair of configurations. And for a given meiosis, right, there are two configurations. So you could have, this with this one. So this would be one pair that would happen as an outcome of one configuration of meiosis, big R with big Y, segregating away from little r with little y. And then this would be the alternate type of pattern that could happen, and you end up with a 1/4 chance for each one. Okay, so one thing to keep in mind is that this is like with two different genes, but you don't have to you could ignore one of the genes, right? And you could just say, okay, within that set of probabilities, what's the probability of just getting round to wrinkled, right? So if you wanted to just look in here, you could say, oh, the segregation probability of round to wrinkled. Well, I'm just going to say, okay, well, let's just focus on round. Well, what fraction of this set of sort of 16 are round? Well, 9/16, and 3/16, right? 9/16 are round, and then these 3/16 are round. And then the wrinkled, right, are these 3/16 right here, and this 1/16. So 9/16 plus 3/16 here and here, plus 3/16 and 1/16 here and here, becomes 12/16 to 4/16. That's just 3 to 1. So within that set, you could just focus on one phenotype, and you'll see that 3 to 1 pattern that's happening as you might expect. All right. There's two more final things I want to kind of quickly remind everybody about, is that these patterns of segregation, the sort of the Mendelian patterns, don't always apply to organelles. So, for example, the mitochondrial is inherited only from the mother. And so here is GFP mitochondria, green fluorescent protein mitochondria in an oocyte. It's been labeled that way. And what this means is that the mother here, all right, passes the mitochondria to all her offspring, right? And dad has no contribution. And so if there's a mitochondrial defect, all of their offspring will inherit it, right? But these males won't transmit it to their offspring because only the mother will transmit it. So the female lineage will continue transmitting this defect from here, right? But these males will not contribute that, right? And you can also see those types of patterns in chloroplasts, which are the photosynthetic element within plants. Those are oftentimes inherited, or usually inherited, through the cytoplasm. And so you can actually have interesting patterns through mitosis in which a defective chloroplast can arise and be transmitted through mitotic events. And so you end up with these kind of interesting patterns where you have two different genotypes of a particular allele, and you get what is called cytoplasmic segregation. It's not meiotic. It's just that by random drift between these two different allelic states within a cytoplasmic organelle, you can end up with cells that actually only have one genotype of an organelle and other cells that only have another genotype of an organelle. And you can actually see these types of patterns sometimes in plants, in which a certain lineage, in this particular case, this entire lineage, has inherited through cytoplasmic segregation a set of cells that all have, in this particular case, a defective chloroplast. So it's always important to remember those other types of weird types of inheritance with organelles, but obviously for the most part we're focusing on nuclear inheritance, which is what we associate with Mendel.